Good afternoon. You are welcome to another lecture video on chemistry of lipids. In our previous class, we discussed about phospholipids, and we have seen the various types, classes of phospholipids. We discussed about glycerophospholipids and sphingophospholipids. Under glycerophospholipids, we talk about various types of phospholipids, glycerophospholipids that are found within the cell, in the cell membrane. So in today's lectures, we are going to be discussing about another example of glycerophospholipids, which is phosphatidylglycerol. And we know that glycerophospholipids, generally, they have what phosphatidic acid backbone. So all glycerophospholipids are derivatives of phosphatidic acid. And we say phosphatidic acid, if you look at the structure of glycerol, the first carbon and the second carbon is having fatty acids that are attached in what? Ester linkage. So you have what? Ester linkage in carbon 1 and carbon 2, the glycerol, while at carbon 3, you have a phosphoric acid residue attached to it, and you have your substituent group depending on the type of glycerophospholipid you are talking about. If it is ethanolamine that is attached to the phosphoric acid, you have what? Phosphatidylethanolamine, and so on and so forth. So for phosphatidylglycerol, which is another glycerophospholipid that is found in the mammalian membranes, but is found in low amounts. The structure consists of glycerol backbone that is linked to two fatty acids and a phosphoglycerol molecule. So you have carbon 1 and carbon 2 of the glycerol attached in ester linkage to fatty acid, while the carbon 3, you have phosphoric acid residue attached to the carbon 3. In the phosphoric acid, you have what? You have glycerol, another glycerol attached to it. So that is your phosphatidylglycerol. So by now, you must have assimilated the structure of phosphatidic acid. So it's just the substituent groups that you will be attaching to the oxygen of the phosphatidic acid, and you will be having your different types of glycerophospholipid. And you have already learned the structure of glycerol, so you attach the structure of glycerol to the oxygen in the phosphoric acid residue to have what? Phosphatidylglycerol. So what is the function, or what are the functions of phosphatidylglycerol? We know that it is found in the what? In the cell membrane, and it is the second most abundant lipid that is found in lung surfactants. You know, we said that surfactants are surface active agents, and it is comprising up to 11% of those surfactant lipids that are found in the cell membrane. And it is one of the phospholipid that is used to determine the maturity of babies' lungs. And it's also key intermediate in the biosynthesis of other lipids, but especially cardiolipin, which we are going to be discussing in subsequent classes. So for phosphatidylglycerol, it can also be said to be found in plants' cell membrane, where it helps in what we call photosynthesis. You all know photosynthesis. So it is a key component of the thylakoid membrane that is found in plants and in some algae, where it helps in converting light energy into chemical energy. We have already known that it is the constituent of lung surfactant, which play a major role in protecting the lungs from pathogens and allergens and also harmful particles. It is also found in mitochondrial membrane, where it is serving as precursor for the synthesis of cardiolipin. And it is also serve as what activator of RNA synthesis in mammalian system. And it also helps in the innate immune defense, where it helps protect against lung pathogens, allergens, and harmful particles. So the next class of glycerol phospholipid is phosphatidylglycerol, which is an important phospholipid that is both as membrane constituent and as participant in essential metabolic processes in all plants and animals, both directly and indirectly via its metabolites. It consists of a phosphatidic acid backbone that is linked via phosphate group to a molecule of inositol. Inositol is hexahydroxycyclohexane. So you have phosphatidic acid as the backbone, and it is linked to inositol via phosphate group. So what are the functions of phosphatidylinositol? They are intimately involved in innumerable aspects of membrane trafficking and signaling, especially in eukaryotic cells, and it also functions as an essential molecule in cell growth and metabolism. We have what we call phosphatidylinositol 4,5-bisphosphate, which is an important constituent of cell membrane phospholipids. And when the breaking down of phosphatidylinositol 4,5-bisphosphate occurs, it is cleaved into diacylglycerol and inositol triphosphate. These two molecules, they serve as internal signals or secondary messengers. So we have another example of glycerol phospholipids, which is called diphosphatidylglycerol. And this diphosphatidylglycerol, it is also called cardiolipin. You know, we discussed about phosphatidylglycerol. So now you have two molecules of phosphatidylglycerol joined together to form diphosphatidylglycerol, which is cardiolipin. It is a unique glycerol phospholipid that is having two molecules of phosphatidylglycerol, and it has some important properties that you should take note of. It has 4 acyl groups, 3 chiral centers, and it has 2 negative charges. So it has 4 fatty acid groups, 4 acyl groups. It has 4 chiral center. When we say chiral center from your chemistry, you remember a carbon that is attached to 4 different groups. You know, carbon is tetravalent. So a carbon that is attached to 4 different groups, that carbon is called a chiral carbon or chiral center. So diphosphatidylglycerol or cardiolipin has 4 acyl groups, 3 chiral centers, and 2 negatively charged groups. These 2 negative charge comes as a result of, you know, phosphoric acid residue is having oxygen here, and this oxygen is carrying negative charge. And if you look at it, the first phosphatidylglycerol, and you know it's diphosphatidylglycerol. So if you look at this, you have glycerol backbone here attached to fatty acid in carbon 1 and carbon 2. You have fatty acid. So if you look at carbon 1, it is bonded to fatty acid and bonded to hydrogen and another hydrogen. So this carbon is not a chiral carbon. But carbon 2 here, it is bonded to fatty acid It is bonded to another group here, CH2RCOO, and it is bonded to hydrogen, and also it is bonded to another different group. So this carbon is a chiral center. Another chiral center can be found here. You see this carbon, carbon 2 prime, is having a group here, and it has hydrogen attached to it. It has OH attached to it, and another different substituent group. So it is a chiral center. Another chiral center is this carbon of the glycerol 2B that has this CH2 and fatty acid residue, right? And it is attached to hydrogen and another fatty acid residue and another group here. So that is your 3 different chiral centers, and the negative charges here, -1 and -2. So that is how you comes about the 4 acyl groups and 3 chiral centers with the 2 negatively charged. So cardiolipin is a major lipid of mitochondrial membrane, okay? And the trivial name of cardiolipin is derived from the fact that it was first found in animal hearts, where it is especially abundant, but it can be found in mitochondria of all animal tissues. They are important phospholipids components of membranes that are involved in the generation of electrical potential for energy synthesis. So the next class of phospholipids is plasmalogen. These are lipids that are constituent of as much as 10% of the phospholipids of brain and muscles. So structurally, the plasmalogens resemble phosphatidylethanolamine, but they possess an ether link in the SN1 carbon instead of the ester link that is found in acyl glycerol. So plasmalogens, they have what? An ether link instead of the what? Ester linkage. So if you look at the structure here, typically it is having an alkyl radical in an unsaturated alcohol. And in some instances, you can have choline, serine, inositol, or maybe which may be substituted for what? Ethanolamine. So if you have ethanolamine here, and this is the what? Alkyl radical, that is what? Unsaturated alcohol. So this is the typical structure of what? Plasmalogen. So what is the meaning of this SN1? This is what? Stereo specific numbering, and we are going to be explaining it. In lipid chemistry, the SN system refers to the stereo specific numbering system, in which is used to precisely identify the positions of fatty acid chains on the glycerol molecule, particularly in triglycerides and phospholipids. So by designating the carbon atoms as SN1, SN2, and SN3 based on their stereochemistry, especially indicating which side of the glycerol molecule the fatty acid is attached to. So vertebrate heart tissue is uniquely enriched in ether lipids. About half of the heart phospholipids are plasmalogens. An ether lipid, platelet activating factor, is a potent molecular signal. It is released from the leukocytes called basophil and stimulates platelet aggregation and the release of serotonin, which is a vasoconstrictor from the platelet. It also exerts a variety of effects on liver, smooth muscles, heart, uterine wall, and lung tissues, and it plays an important role in inflammation and allergic response. So we are going to stop here, and we will continue in our next class. Thank you for watching. If you are yet to subscribe to the channel, kindly subscribe and turn on notifications so that whenever we upload a new video, you will be notified. Thank you. See you next.